Hello. Welcome to World of Microbes. In today's video, we are gonna introduce you to diagnostic microbiology, its significance, laboratory procedures used in the diagnosis of infectious diseases, general rules to all specimens. Diagnostic microbiology is a laboratory science that analyzes samples from humans, animals, or the environment to identify pathogens and diagnose infectious diseases. It also helps treat patients by identifying and quantifying pathogens. Since the discovery of the germ theory of disease, scientists have been finding ways to harvest specific organisms. Using method media or genome sequencing, physicians and scientists can observe novel functions in organisms for more effective and accurate diagnosis of organisms. Diagnostic microbiology has been developing to rapidly detect and accurately identify implicated microorganisms in test specimens through a variety of techniques. Technologic changes have made constant and enormous progress in the various areas including bacteriology, mycology, mycobacteriology, parasitology, and virology during the past 2 decades in the field of microbiology. The field of diagnostic microbiology determines whether suspected pathogenic microorganisms are present in test specimens collected from human beings, animals, and environment, and if they are present, identifies them. In medical practice, a fundamental principle of diagnostic microbiology is to define infectious processes and explain treatment options through rapid detection of specific pathogens. Thus, beyond detection, quantitation, and identification of microorganisms in clinical specimens, diagnostic microbiology also provides physicians with antimicrobial susceptibility profiles of the identified microorganism. Advanced diagnostic techniques also provide tools in basic microbial pathogenesis studies. Diagnostic medical concerned with the etiologic diagnosis of infection. Do like and subscribe. A microbiology laboratory is a laboratory devoted to the culturing, examination, and identification of microorganisms including bacteria, fungi, yeasts, etc. The microbiology laboratory has a crucial role in effective infection prevention and control, IPC. The job of the clinical microbiology laboratory include to test patient specimens for microorganisms. A healthcare professional collects a sample, like blood, urine, sputum, or tissue, from the suspected infection site and sends it to a laboratory where technicians use techniques like bacterial culture to grow and identify the present microorganisms, often utilizing microscopy to visualize them and further analysis to determine the specific species. This process helps diagnose the infection and guide treatment decisions. Do like and subscribe. To provide information, when appropriate, about the in vitro activity of antimicrobial drugs, the ability of an antimicrobial agent to inhibit or kill microorganisms in a laboratory setting, typically measured by testing its effectiveness against a specific bacteria or fungus in a controlled environment, like a petri dish, without involving a living organism. Essentially, it's how well the drug microbes outside the body. Do like and subscribe. Confirming a clinical diagnosis of infectious disease with a bacterial etiology, which is the study of the causative agent of a bacterial infection. Do like and subscribe. To advise the physician as well as process specimens, which is the process of preparing biological samples for analysis. It involves obtaining the sample, treating it, and then analyzing it. Do like and subscribe. The clinical microbiologist should participate in decisions regarding the microbiologic diagnostic studies to be performed. The most appropriate study depends on the suspected disease, the available tests, and the type of specimen collected. Do like and above all, the clinical microbiology laboratory, whenever appropriate, should provide an interpretation of laboratory results. Interpreting laboratory results involves comparing a patient's test values to a normal reference range provided by the lab, where a result falling within that range is considered normal, while values outside the range may indicate a potential medical condition requiring further. This analysis takes into account factors like the specific test performed, patient history, and other clinical findings to reach a diagnosis. Do like and subscribe. Laboratory procedures are a set of safety and health standards that ensure the safe use of equipment and chemicals in a laboratory. Laboratory procedures used in the diagnosis of infectious diseases are as follows. Do like and specimen selection, collection, and processing. A properly collected specimen is the single most important step in the diagnosis of any disease. This is because the results of diagnostic tests for infectious diseases depend upon selection, timing, and method of collection of specimens. The specimen must be collected from the anatomical site most likely to yield the agent at that particular stage of illness. The specimen must be handled in such a way that will favor the organism's survival and growth. Do like and subscribe. No. 1 is morphologic identification of the agent in strains, specimens, or tissues, that is identifying a microorganism within a strain by observing its physical characteristics, such as its shape, size, arrangement, and other visible features under a microscope, often using staining techniques to enhance visualization and differentiate between different types of microbes. This method is the initial step in identifying a bacterial or fungal strain before further molecular analysis. Using light and electron microscopy. Do like and subscribe. Culture isolation, the process of separating and extracting a single type of microorganism from a mixed population, allowing for the study of its unique characteristics by creating a pure culture where only one specific microbe is growing, achieved through techniques like streak plating or pore plating on agar media, and identification of the agent after isolating and separating the specific microorganism. Detection of antigen from the agent by immunologic assay. An immunologic assay, also known as an immunoassay, is a biochemical test that measures the amount of a substance in a solution. It uses an antibody or antigen to detect the presence of a molecule, called an analyte. Immunoassays can detect the presence of disease-causing bacteria and viruses in blood or urine. Immunoassays the activity of genes, cell proliferation, and drug effects. Do like and subscribe. The polymerase chain reaction, PCR, technique is a method for making copies of a DNA sample. It's used to amplify small amounts of DNA so that scientists can study it in detail. Do like and Antibody demonstrations are techniques that show the presence of antibodies in cells or tissues. These techniques use immunological reactions and fluorescent labeling to identify antibodies. Do like and The quantity of specimen must be adequate. The quantity of a specimen depends on the type of specimen and the test being performed. The quality of the specimen is also important, as it affects the accuracy of the test results. The quality of the specimen depends on how and when it was collected, and how it was preserved and transported. Improper collection can lead to misleading diagnoses and potential danger to the patient. It should be representative of infectious process, that is a specimen collected from a part of the body where an infection is suspected, such as a throat swab, urine sample, blood draw, wound swab, spinal fluid, from a lumbar puncture, stool sample, or a tissue biopsy, depending on the suspected site of infection. These samples are then analyzed in a laboratory to identify the specific pathogen causing the infection. Avoiding specimen contamination by using only sterile containers and observing aseptic precaution. Wear protective equipment, and clean surfaces regularly. Do like and Lab specimens should be transported to the lab as soon as possible after collection to prevent delays in testing. Delays can change the values of the analytes being measured. Do like and Specimens obtained through operation require special attention. Enough tissue must be obtained for both histopathologic and microbiologic examination. Histopathologic examination is used to distinguish neoplastic from inflammatory lesions and acute from chronic inflammation. The type of inflammation present can guide the type of microbiologic examination performed. If, for example, granuloma is observed histopathologically, microbiologic examination should include cultures for mycobacteria and fungi. Do like and